Welcome back to our Bio180 lecture series. Today, we're going to be continuing with our exploration of Unit 4, cell metabolism. And so as a brief review, we've already gone through what we called our biochemistry boot camp. And then we have been working on the various reactions of cell respiration. So we've gone through glycolysis. And then in our last lecture, we went through the citric acid cycle. Today we're going to complete our tour of cell respiration by covering the electron transport chain and oxidative phosphorylation. Later on, we'll go over our final pathway, which is going to be photosynthesis. And this will be an example of an anabolic pathway. So right now we're going over a catabolic pathway. So, as a brief overview, what we're going to be doing today, remember from the end of our citric acid cycle, we have produced a lot of what we called reduced coenzymes. So specifically, and we even produced some in glycolysis, we talked about producing them in beta oxidation. So we have a lot of NADH, and then we have a few molecules of FADH2. And so these are the reduced form. They have the hydrogen on there, but more importantly, they've got a couple of high energy electrons that are being carried by these coenzymes. They're acting as a kind of electron shuttle. And so what we're going to be doing today, as a brief overview, is essentially we're going to be creating a battery inside that cell, and we'll show you how this works. And we'll have a battery, and that battery is going to be used to power a pump, and then we're going to get energy out of it using sort of like a water wheel approach. And so these are the analogies that we're going to be using as we look at the electron transport chain. So first, let's attack this idea of a battery. If you look at a very simple battery, this isn't one you buy at a store, but a very simple battery. To have a battery, you have two chambers, and in one chamber, you have to have an electrode that is willing to easily give up electrons. And so as just an example, if we had one electrode that was pure sodium metal, at the beginning of the class, we talked about how sodium likes to give up electrons to reach its complete octet. And so if we had sodium on it, and then inside this battery, we'll also have sodium and then sodium ion. And as the reaction proceeds forth, we're going to get the sodium converted into sodium ion. And it does so by giving up an electron. And then on the other side of the battery, we're going to have a electrode right here. And in this electrode, we're going to put some chlorine gas. Chlorine, remember, as an element, likes to receive electrons. It's an electron sucker. And so it likes to pull electrons. And when it pulls those electrons, of course, it forms chloride ions. So the chlorine will convert into chloride ion. And basically what a battery will always have these two different electrodes. And then if I put a wire between them so that I have a pathway for those electrons to run, the electrons will actually jump off of the sodium, run through the wire, and then jump onto the chlorine, and that produces our chloride ion. And the movement of electrons then is what we call electricity. And so that's how a basic battery, well, you can have different elements and chemicals as the two electrodes, but you always have to have an electron donor, something that easily gives up electrons, and you have to have an electron sucker, something that pulls electrons toward it that has a high electronegativity. And that's what we're going to actually see today. Our electron donors are going to be NADH and FADH2. So they're actually, I could replace this electrode in the cell with NADH. It's going to provide electrons, and then it becomes NAD plus on this side. And those electrons are going to flow through my electron transport chain. And we also need, we're not going to use chlorine. The living systems don't use chlorine because that would be too toxic. Chlorine doesn't seem to do well for electron systems. So instead, we are going to use another element that's really good at pulling electrons, and that's going to be oxygen. So oxygen then is going to be our electron sucker, and oxygen is going to form, we'll talk about this a little bit later, but it's going to form water, H2O. And we'll talk about that different oxidation step when we get there. But basically what we're setting up inside the cell is a simple battery. We're going to donate electrons from NADH. They're going to flow through the electron transport chain. And just as a battery is there to provide electricity, provide power, we are going to harness this electron flow and use it to power a series of pumps inside the electron transport chain. So we'll have some different complexes instead of just a straight wire here. We are going to have some pumps, like water pumps, like you might find in a fish tank or in a farm somewhere that moves water. But instead of moving water, we are going to move hydrogen ions. And so we're going to create a hydrogen ion concentration gradient. And then we're going to use those hydrogen ions to flow like water flows through a water wheel to spin it and generate power. We're going to see these hydrogens flow through a kind of molecular water wheel called ATP synthase in the membrane. And then that water wheel is ultimately going to produce ATP, and that's what we're trying to get out of the whole process. So that's a brief overview of what we're looking at and sort of an analogy of how to think of these things. So now let's move into the electron transport chain itself and what it looks like and how we're going to get some energy out of it. To draw the electron transport chain, we're going to start with a series of 3 rectangles. So we're going to draw these 3 rectangles. And then we're going to have a membrane that's going to go through these. And these 3 rectangles are going to be our pumps that we talked about earlier. And so they actually have names, but we're just going to call them complex 1 for right now. This is going to be complex 3, and this is going to be complex 4. And so these again are going to be pumps. Their goal or their job is going to be to take hydrogen ions from one side of the membrane and pump them to the other side of the membrane. So let's go ahead and put each one of those pumping those hydrogen ions. And let's go ahead and label. So which membrane is this? This is the mitochondrial inner membrane. Inner membrane. And then the 2 spaces up here, we actually have what's called the intermembrane space. And then down here on this side, this is going to be the matrix. So that kind of orients us to where we are. There are a couple other players we want to draw. For example, you may have noticed I went 1, and then it looks like I skipped over 2 and went to complex 3 and complex 4. There is a complex 2. It turns out to not be a pump. Complex 2 is located right here. And it's going to be the entry point for F80H2. We'll talk about that in a second. Other things that we need to show up here. We have these complexes, and what's going to happen is electrons are going to move through these complexes, like they might move through a wire. But we also need to be able to get the electrons from complex 1 to complex 3. So we need some kind of shuttle. These are big, bulky complexes. We don't want them crashing into each other. And so we need some kind of smaller intermediate shuttle. And so I'm going to put a little dot right here. And that's one of the shuttles. You have this shuttle, it's called ubiquinone. So usually we just write it as a UB in a diagram like that. And we'll write over here. UB equals ubiquinone. And so ubiquinone is kind of unique in this process in that ubiquinone is a lipid. So it's not a protein. So it's a lipid, small molecule. And it's an electron carrier, or an electron shuttle. So this is actually another type of cofactor. We've talked about cofactors before. Ubiquinone is another type of cofactor. And it resides in the membrane, and it's going to take our electrons from complex 1 and shuttle them over to complex 2. We also need a shuttle between complex 3, shuttles from complex 1 to complex 3. We're also going to need a shuttle between complex 3 and complex 4. And this is actually a peripheral membrane protein called cytochrome C. And so cytochrome C is the shuttle between complex 3 and complex 4. And then we have one final piece of this process, one final protein that we need to add, and that is going to be this enzyme called ATP synthase. Okay, so those are the players in our process. This is sort of the stage of our production and the different actors that are in it. So the stage, again, is the mitochondrial membrane, the intermembrane space, and the matrix are the different areas. And then we have these different players involved in the process. Now, the production is something like this. So we're going to start out here in the matrix. And we were created in the citric acid cycle, we've created a whole bunch of this NADH. And so we're going to have NADH, and it's going to come up and it's going to donate its electrons and thereby create NAD+. And at the same time, we're just going to create NAD+, and we're going to dump off those 2 electrons. Now, those 2 electrons are going to jump around in here. There's some different cofactors inside of this that the enzymes are jumping from one cofactor to another until they eventually end up moving over there to ubiquinone. And as these electrons jump around to this various cofactors in complex 1, that movement of electrons is what pumps those hydrogens across. Okay? And so that's important to understand. Overall, if I pump 2 electrons through this whole process, I'm going to get about 4 protons that get pumped through. Now, the electrons then are going to hop onto ubiquinone, And from ubiquinone, they get shuttled over into complex 3. And in complex 3, again, we move through different cofactors in complex 3. And as we move through complex 3, we again are going to pump. Again, it's about 4, depending on who you talk to, about 4 protons that we're going to get pumped. As the 2 electrons move through, they're going to move about 4 protons across again. And then cytochrome C is going to shuttle our electrons from complex 3 over to complex 4. In complex 4, they are again going to hop between different cofactors embedded in that complex until they end up right here. And now that the electrons have been moving through, so this is the electricity part, as the electrons are moving through the different complexes, they're providing electricity, quote unquote, and that's what's powering these pumps to move these protons against their concentration gradient. And so now what happens when we hit complex 4? Well, this is where oxygen comes into play. So we're going to take an oxygen molecule, O2, and we're going to come through and pick up those electrons. We're actually going to, in this case, for right now, we'll say we're going to take half of an oxygen, is the way you'll often see it written in the literature. And then we're going to go pick up those 2 electrons, and that's going to create, again, if we split this and it's neutral, and then we add 2 electrons to it, we create O2 negative. So that's an oxygen atom with a -2 charge on it. That's not very stable, so we have a couple of protons that get attracted to that -2 charge, and we end up just creating a molecule of water. And so we're going to take O2 and turn it into water. And I want to take a second and show you a little bit about how this works and how water or oxygen is able to absorb or attract those electrons. So if we take a molecule of water, or of oxygen, has a double bond, looks like this, and I'm going to redraw it as a Lewis diagram. And so in our Lewis diagram, we would have our 8 electrons, 2 pairs are shared, and so that's the Lewis diagram of molecular water. And so one of the first things that we do is we split that water molecule. We're going to split this bond, so we're just going to move and we're going to have each oxygen atom as it pulls apart. So notice that right now we've kind of split these, but now this oxygen atom does not have its octet of electrons. It's deficient. So we need to add, and those are the electrons that are going to come from complex 3. So we're going to have 2 electrons that are going to come from complex 3, and they're going to fill that to make an octet. And that's how we're getting the oxygen. But when they do that, with the 6 electrons in the outer shell, it was neutral, it had no charge. And then when you add these 2 electrons, that's how it gets its -2 charge. And then we're going to just bring along a couple of hydrogens. Remember, these are ions, hydrogen plus, and so they don't have any electrons. So they are just going to hop on. Another one hops over here, and that's how we get water. The other oxygen also has to pull electrons, and so in order to split one oxygen atom with, or one molecule of oxygen with its 2 oxygen atoms, we actually need 4 electrons. 2 of them we got from NADH. And so if we wanted to get another 2 electrons to complete our molecular oxygen to water transformation, we could go ahead and run a second NADH through. That would be fine. So we could run 2 NADHs through. However, let's use this for a moment. Another thing that we can do is take an FADH2. And if we run FADH2 through complex 2, then we create an FAD. And we get another 2 electrons. And those go right into complex 3. So they'll feed in right there. They'll pump some protons through complex 3 and complex 4 and give me another 2 electrons that I could add to my molecular oxygen. Okay? So, in summary, we can run both NADH through complex 1 and then 3 and 4, or we can run an FADH2 through complexes 2, 3, and 4. And that's how we're going to get our electrons to make water. And that's also how we get the energy as the electrons flow to pump those protons. That's the electron transport chain. And that's how it works. The primary driver of this is right here, the electronegativity of oxygen. That's why we have to continually breathe in oxygen. We have to take that oxygen to add to complex 4 as an electron sucker. So this whole thing is being drawn by the electronegative power of oxygen. And that's how we get our energy. Now notice through this process, the electron transport chain does not produce any ATP. All we've done, we have used our electron energy, but what we've really done is we've created these protons, and we've created a proton gradient on the inner membrane space. Right here, this last one is about 4 and 4. And so we call this the proton motive force, but we've developed this proton gradient. And so we've got a lot of protons on this side. There's an imbalance of protons. There's a gradient. And so the protons naturally want to return to the balance, equilibrium. And so protons would like to go across the membrane. Just like water, if you build up water in a dam, if you build a dam and there's a bunch of water on one side, if you provide a hole, that water will just naturally flow through the dam. And if we provide some kind of turbine for that, or water wheel for that water to flow through, then the flow of water will actually power that turbine. Well, that's what we're going to do in this case. We're going to take this proton gradient, and we're going to provide a hole in the membrane. The membrane is sort of like our dam. And we're going to provide a hole in the membrane in the form of ATP synthase. And so what happens in ATP synthase is that our extra protons are going to run through. ATP synthase has several different parts to it. One of the parts, this is called the F sub 0. This is like a carousel up in here. So in the membrane, you've got about 10 to 12 of these smaller subunits that are sitting in the membrane. And they act as a carousel. Protons will come in, land on one side, and cause, as they pass through the membrane, they cause this carousel to spin, just like a merry-go-round. And so in the membrane, this carousel is going to spin. As that carousel spins, it spins this central shaft right here in the middle. And then at the bottom, we have these catalytic subunits. So you've got alpha subunits and you've got beta subunits. And you've got 3 pairs of those alpha beta subunits. And this is where there's a binding site right in the middle of those alpha beta subunits. And this is where we're going to take ADP and we're going to add a phosphate, and those are going to enter into that site, and then out of it are going to come ATP. And so the way this works is there's a conformational shift that has to happen in these alpha and beta subunits. And so it works kind of like if you can imagine my body being the alpha and beta subunit. So there's an active site, and let's pretend that this pen is a molecule of ADP, and then the lid represents the phosphate, the inorganic phosphate. And so what happens is ADP will come in and land in one side of the active site, and the phosphate lands in the other. And then this shaft, as it spins, has kind of a protrusion on the back of it. And so if I'm one of the alpha-beta triplets, as that protrusion comes and hits me in the back because of the spinning carousel, it causes a conformational shift, and I'm going to take that phosphate and that ADP and slam them together, and that's how I form ATP. And so that one's formed and done, and then there would be another ADP that would come in, another phosphate, and I would hold it and wait until that spindle came, that protrusion came and hit me in the back again. And as soon as it hit me, I would slam these together, and then when it released, I would release that ATP, and then I would grab another one. So as the carousel spins, the shaft will hit, in turn, those different alpha-beta pairs, and they will slam the ATP together, release, grab, slam, release, grab, slam, release, and it would just go through that reiteration over and over again. And that's how ATP synthase is going to make the ATP that we need to get out of the electron transport chain. This is how the majority of the ATP in your cells is formed. Hydrogen ions or protons flow through the carousel. They make the carousel spin. The carousel makes the shaft spin. The shaft hits the alternating alpha-beta pairs and causes them to crash a phosphate and an ADP together, making ATP. So that's the overview of the electron transport chain and ATP synthase, or oxidative phosphorylation. Now, let's talk about a couple things. If we come over here, every time we run an NADH, let's talk about what we call stoichiometry. Stoichiometry is the relative amounts that we produce in this process. Now, a couple of rules here. Every time we put one NADH through the electron transport chain, so realize that one NADH produces 2 electrons. And those 2 electrons, as they move through complex 1, 3, and 4, produce about 10 to 12 protons pumped into the inner membrane space. Does that make sense? Because we're going all through, we have 4 plus 4 plus 4. Sometimes we lose a little, or they're not quite as efficient, and so the actual stoichiometry is somewhere between 10 and 12. Now, it turns out that 10 to 12 protons, as they flow through, so as 12 protons flow through, that produces 1 full turn of that carousel. 1 full turn of the F sub 0 region of ATP synthase. And 1 full turn will produce, 1 full turn means that that shaft goes through all 1 full turn also. So it hits the first alpha beta pair, it hits the second alpha beta pair, and it hits the third alpha beta pair. So that means I'll produce 3 ATPs with 1 full turn. So then I get 3 ATPs produced. Okay? And that's what we really want to get to. So some of this stoichiometry is just getting to this 3 ATPs. We could do the same analysis with FADH2. So if I have one FADH2, again, 2 electrons. However, with FADH2, my electrons, they don't go through complex 1. FADH2, remember, has a lower power. It's like a AAA battery instead of a AA battery. And so it doesn't have enough electron power to donate to complex 1. It's lower in energy. So it can only donate its electrons to complex 3. That means those electrons are only pumping in complex 3 and 4. So we don't get as many protons being pumped when we use FADH2 as an energy source. So now we're only getting about 8 protons pumped. And so now we only go through about 2/3 of a full turn of ATP synthase. And since we only go through 2/3, so up here this would be 3/3, and so we produce 3 ATP. 2/3 produces only 2 ATP produced. And so we're only going to get about 2 ATPs out of our FADH2s when we run them through electron transport chain. Generally, these numbers right here are what you'll see in biology textbooks or what we as biologists talk about. If you get into biochemistry or you get into a chemistry class, you'll see that they reduce these a little bit, and they'll say about 2.5 ATP and about 1.5 ATPs. And that's just a little bit more accurate representation. Remember, we're creating a proton gradient. Not all of these protons will actually flow through the ATP synthase. Some of them actually are used in some other transport processes. And so this 3 and 2 represents a whole number, sort of maximum, and this is the more physiological average. So that's a little bit about how many ATPs we're going to get out of NADH and FADH2 in this process. Now, let's come back over here to our beginning, and let's summarize. Now that we've been through that, so that's the entire process of cell respiration. Let's go back and look at some of our totals of ATP that we might get out of this process. Remember, cell respiration included a couple different processes. We started with glycolysis. And so where did our energy go from glycolysis? Well, we had the potential to produce some ATP and then some NADH. And this is what we'll track. We'll track the FADH2s also. So in glycolysis, you remember we had a net production of 2 ATPs. And then we also got 2 NADHs out of it. And then we got 0 FADH2s. Then we had that small process called pyruvate transport. Pyruvate transport. We didn't get any ATPs out of pyruvate transport, but we did get 2 NADHs. And by the way, this is going to be for, we're going to look at this and say this was for 1 glucose molecule, because we had to start glycolysis with 1 glucose molecule. So out of pyruvate transport, that's what we got. We ran the citric acid cycle. So out of the citric acid cycle, we're going to actually, per glucose, we have to run the citric acid cycle twice around because we had 2 acetyl-CoAs. So we're going to get 2 ATPs out of the citric acid cycle. We're going to get a total of 6 NADHs as we run that through twice. And here's where we get our first. We got 2 FADH2s. Now we run the electron transport chain and oxidative phosphorylation. So we'll call it electron transport chain, oxidative phosphorylation. Here we get a big yield of ATP. We're going to take these. So right now we've got 10 of these NADHs. So let's subtotal that 10. We've got 2. So we're going to take this 10, and out of electron transport chain, we're going to get 3 NADH, or 3 ATPs for every NADH that we put into it. So if we take 10 times 3, we're going to get 30 ATPs out of our NADHs. Remember I said that those NADHs hold a lot of power, and that's where we're going to get a lot of our ATP from. And then our FADH2s, we're going to take that 2 and multiply it by 2 ATPs. So we're going to get a total of 4. So over this whole process then, we add up the number of ATPs that we get, and the maximum total is about 38 ATPs per glucose molecule. Okay? Now, there is a little bit of a discrepancy. When you look in your textbook or consult online, you'll see that they'll often write 36 to 38 ATPs as the maximum yield of cell respiration using glucose. Why is that? Right here, it has to do with these 2 NADHs. Remember, these 2 NADHs were produced actually in the cytosol, but over here, ATP synthase is working in the matrix. And so in some pathways, the transport of those NADHs into the matrix is a reduction. We lose a little bit of power, and instead of making NADH, that transport tends to convert them into FADH2s. And so we'll lose a couple of ATP out of it due to transport. And so sometimes you'll see 36 to 38. This is if we transport it in one pathway, another pathway, you are able to conserve all the energy. So you often see 36 to 38 ATP total. And that, by and large, is cell respiration for us. One more thing we might want to do before we end is let's come back over here. We just went through glycolysis and how that works. But remember, we went through some other pathways, including beta oxidation. And so let's do a summary. We did glycolysis. Let's do a beta oxidation summary too. So remember, in beta oxidation, we're going to take a fatty acid. Now we're actually taking a fat, but I'm just going to break it down. I'm going to start with a fatty acid. And so if we start with a fatty acid, let's make it an average size for a fatty acid would be 18 carbons long. So we're going to do an 18 carbon chain. So let's put our processes here together. Remember, for beta oxidation, we're not going to use glycolysis, but we're going to start with beta oxidation. That's our first pathway. So we got beta oxidation. We're going to have the citric acid cycle. There's no pyruvate bridge in this. And then we're going to have electron transport and oxidative phosphorylation with ATP synthase. Now, up here, we're going to track it a little bit differently. We do want to track, let's redraw this. We do want to know the number of FA, let's see, ATPs we want to know. We want to know FA or NADHs. And FADH2s. And then over here, I'm going to put the number of acetyl-CoAs. That'll be important in a second. So in beta oxidation, we've got a fatty acid. It's 18 carbons long. We taught you how to predict the number of acetyl-CoAs. You just take the number of carbons and divide it by 2. So that means we're going to create 9 acetyl-CoAs out of that fatty acid. In beta oxidation, we don't produce any ATP directly, so that's going to be a wash. But we do produce some NADHs. How many NADHs do we get? Well, we get 1 for every acetyl-CoA we produce except for the first one. So we would get a total then of 8 NADHs out of breaking down that 18 carbon fatty acid. And then we would get 8 FADH2s as well. Now we come to citric acid cycle. When we did glycolysis and cell respiration over there, we ran twice through the citric acid cycle. But now with beta oxidation, each acetyl-CoA goes through the citric acid cycle. So we're actually going to run this 9 times. If we run it 9 times, I'm going to get 9 ATPs. There's that ATP produced at the bottom of the citric acid cycle for every cycle. I get 3 NADHs for every cycle. So now I take 9 times 3, and I get 27 NADHs. And then FADH2s, I get 9 again. Now we come down here to the electron transport chain. I have to add these up. So 8 27 plus 27 is going to be 35 NADHs. I've got 8 and 9, so I'm going to get 17 total FADH2s. So now when I run these through the electron transport chain, I get 3 ATPs for every NADH. That is a staggering 105 ATPs plus another 34 from my FADH2s. So over here, by breaking down one fatty acid, I'm going to get, let's see, that would be 139. So 100, and if I've done the math correctly, about 148 ATPs out of one fatty acid. So before we got 38 ATPs out of burning sugar, and out of burning a single fatty acid, I get 148 ATPs. Now there's a couple reasons for that. This particular fatty acid, it has 18 carbons in it. That's 3 times as long as our 6-carbon sugar. So we're doing a lot more oxidation of these 18 carbons, and that's why we're getting more ATP out of it. But notice, even for that, this would be approximately 50 per 6-carbon units, right? We're still getting more ATP out of our fatty acid than we did out of our glucose. Why is that? Remember that our glucose, we're getting our ATP out of it because we're doing all these oxidation reactions. So fatty acid is becoming oxidized to CO2. That produces our reduced coenzymes by oxidation reduction reactions. So the more oxidations I do, the more reduction products I get, the more ATP I can get. Remember that glucose already starts pretty highly oxidized. It's already got a lot of oxygens on it. Fatty acids have relatively few oxygens on it. So more oxidations, more reductions, more ATP. And that's why fats will often be a better source of energy. If you really need ATP energy, you're better off eating fats than you are sugars because you can get more ATPs out of it. So That's a summary of beta oxidation. We've got a summary of cell respiration using glucose, and then we've gone over the electron transport chain and our oxidative phosphorylation. Let's come back to one more point over here with glucose. These ATPs right here were made through this process called oxidative phosphorylation, meaning that these ATPs were made using ATP synthase and the electron transport chain. That's one way of making ATP. Notice that these ATPs up here were not. These ATPs were made using what we called substrate level phosphorylation. So, we have then two different ways of making ATP. We can take a high energy substrate and donate a phosphate to make our ATP, or we can come down here and use electron energy to power ATP synthase to kind of slam that phosphate onto ADP and make ATP synthase. So we've got two different methods of making ATP in the cell, substrate level and then oxidative phosphorylation. With that, we'll conclude our tour of cell respiration. When we come back next time, we're going to look at an anabolic pathway. We're going to look at photosynthesis and how plants are able to take energy from the sun and light and use that to essentially run cell respiration in reverse. We're going to take we're going to take carbon dioxide and water, and we're going to combine them to now make glucose and sugar. And so that'll be a really interesting set of pathways to look at and how we make glucose out of really just sunlight and thin air. So we'll see you next time on the Bio 180 lecture series.